is Dr. Sarah Pinney, and she is a pediatric endocrinologist and did her training here at the Children's Hospital of Philadelphia in pediatric endocrinology, and has been working in this area, both research-wise, for many years, starting back when she first developed an interest in the effects of PFAS and in pregnancy. And so today she's going to talk to us a bit about her research, and I think we can get started, Sarah.
Well, welcome everybody. Thanks for joining us for our celebration of Children's Environmental Health Day. And as Becky said, I'm a pediatric endocrinologist, so some of the clinical conditions we're going to talk about today I take care of in our clinic, and I've developed an interest in this area basically because I think that there's a lot of need for more research in this area because we don't purely understand how these exposures to PFAS chemicals and other environmental chemicals are affecting the development of metabolic disease in our children. So with that, I'll go ahead and get started. Let me move here. Great. So I'm sure many of you are familiar with this slide on the left, but I see lots and lots of children who come to clinic due to concerns about obesity. And over the first 2 decades of this century, we've really seen an explosion of the obesity epidemic. There might be a little bit of data that's slowing down, but I think with the pandemic, things have picked up again. But what the slide on the right here, this is a relatively recent publication in the Lancet Diabetes and Endocrinology, has shown that paralleling the obesity epidemic has led to an increase in the diagnosis and incidence of type 2 diabetes in children. And interestingly enough, this has been mostly affected not in non-Hispanic white population, but really in both the American Indian population and non-Hispanic Blacks, which are this red and teal blue lines you can see here where the incidence is really quite taking off. So this is really a strong interest in our research at CHOP, and I think specifically looking at environmental chemical exposure is going to be key to understanding this increase in incidence of type 2 diabetes. The obesogen hypothesis is a hypothesis basically that we know that the obesity and metabolic syndrome is due to many different factors, and it's a very complex endocrine disease. But with that, there's interactions of both genes and environment that are contributing. And as we just saw, there is this abrupt and rapid increase in the obesity prevalence since the 2000s. And we do acknowledge that sedentary lifestyles and high calorie foods are contributing. But with that, we think that there are additional risk factors, including the environment, based on the fact that there's both a geographic disparity of obesity. We also saw that certain populations are affected, and the ubiquitous presence of these environmental chemicals that can certainly promote adipose tissue accumulation in children. So my lab is really interested in looking at how the programming of obesity, diabetes, and metabolic syndrome. And specifically, we know that certain cells are very susceptible to environmental exposures at different stages of a development. So specifically, pancreatic beta cells, adipocytes, hepatocytes are of interest in our lab. Others are studying gametes in the hypothalamus and immune cells. We have looked a lot at exposures such as maternal gestational diabetes and maternal obesity in combination of the fact that environmental exposures are happening throughout the lifespan. So although we're focusing today mostly on pregnancy and gestation and then what happens to the offspring, clearly there are different windows of susceptibility through different ages of development. So the PFAS chemicals, I know many people have already learned quite a bit about them because they've been in the news a lot over the past several years, but really they're man-made industrial chemicals that have both water-repelling properties and they're prevalent in nonstick cookware, also in your water-repellent clothing and stain-resistant fabrics and carpets. One of the main exposures that we've all had is through the water table And a lot of that has been contaminated through firefighting foams, which are prevalent at both military bases and airports. Recently, they have developed some non-fluorinated firefighting foams, but the fluorine carbon bonds in these foams and in the whole chemical substances themselves makes them very resistant to breakdown. And that's partially what gives them the name the forever chemicals. Other exposure can happen through cosmetics, food packaging, and as we just mentioned, drinking water sources, likely through environmental contamination, both through industry and then through military bases and airports. Some of the most common chemicals that have been studied include the sort of base chemicals of PFOA and PFAS, but now there's up to 5,000 to 10,000 different versions of these chemicals, and to the point where it's very difficult to keep track of them all. In terms of environmental exposures and development, these chemicals have very long half-lives in humans, which means that the exposure is persistent for many, many years. So for example, some of the chemicals have a half-life of 3 to 5 years in humans. I actually just was able to update this slide. So here we can see these are mapping the PFAS contamination crisis. Just a few years ago, there were many fewer dots, but you can see here the teal blue here is drinking water with PFAS exposure that's above the proposed limits, either by the EPA or the Environmental Working Group. And as you can see, the entire East Coast is now pretty much covered in above levels. The purple, I apologize here, I'm having some trouble. The purple is the military sites that we know have exposures. And then the darker blue is actually drinking water where they've measured PFAS chemicals, but they've been below the proposed limit. And then there's some other contamination sites. But this was just updated in August. Just a few years ago, there were many, many less sites mapped. And I think this really attributes not that the exposure has gone up in the past few years, but really the level of awareness and measurement has certainly gone up. And now we see that the exposure seems to be ubiquitous. What are the health effects of PFAS? Well, for many, many years, we've documented that they can lead to abnormalities in cholesterol metabolism, both in children and adults. They have primary effects on the liver and liver enzymes. In animals, they can lead to liver tumors, although we haven't really seen that so much in humans. There is some strong epidemiological evidence that they're associated with obesity and insulin resistance, and also epidemiological evidence suggesting they contribute to both gestational diabetes and preeclampsia, but these studies have not shown consistent results. There is good evidence that the PFAS chemicals have strong effects on the immune response and can also affect thyroid hormone production and metabolism. And this was in a nice review by Blake et al. In toxicology a few years ago, but it really is a good figure to describe not only the ubiquity of the exposure, but that how once these chemicals with their long half-lives get into the ecosystem, that they're very hard to remove. So here you can see the industrial sites a lot through the East Coast are aligned with the PFAS contamination areas that are above detection level. On this map, you can see the military fire training areas, and here you can see airports. And so essentially through industry and the aqueous firefighting foams, through military bases and airports, these exposures have gotten into not only the waste infrastructure, but also the water table, which ultimately affects human exposure. And so with all this, remediation effects are underway, but given the long half-life of these chemicals, it's very difficult to remove these exposures from our environment. And just in the past few years, as I alluded to earlier, there's been a lot more press regarding these chemicals and effects on health. So even many of you have probably seen the film Dark Waters. That has to do with contamination from the DuPont plant in West Virginia and effects on exposure on the Ohio River. But just recently, the government, both the EPA and the Biden-Harris administration, have made some moves to help institute national drinking water standards for PFAS and also other regulations. So it's interesting to see that some of the work that's done in the labs to help raise awareness for the health effects of these chemicals has made it into policy. So getting back to our model of programming, fetal programming of obesity, diabetes, and metabolic syndrome, I want to describe first our work setting up an animal model here with the PPAR, the humanized PPAR mouse. So PPAR alpha is a gene that is a fat metabolism gene. It works strongly in the liver, also some in the adipose tissue. And this particular mouse has a humanized version of this gene, since the rodents and human PPAR alpha tend to metabolize fat slightly differently. So in this model, pregnant mice were then exposed to different doses of PFOA to represent a general population exposure through drinking water. And then we assessed the pregnant mouse's glucose metabolism through a glucose tolerance test right before delivery on GD18, so at 18 days of pregnancy. And then we also let these mice deliver and then assessed mom's glucose metabolism after she delivered at both 3 and 6 months postpartum, representing sort of after delivery, when women who have gestational diabetes may go on to develop type 2 diabetes. And then we also assessed some effects from the offspring at 3 and 6 months of age. So here's some data looking at glucose metabolism in the pregnant mice at 18 days of gestation. And here's the glucose tolerance test data. The purple are the mice that received low-dose exposure to PFOA through drinking water. And they had basically a mild form of gestational diabetes with higher glucoses. This is the glucose under the curve, as well as higher fasting glucoses and higher insulin levels. We also found that the liver weights were increased. That's a previously described effect from PFOA exposure during pregnancy. And these mice that were exposed to PFOA had increased fat mass compared to the mice that were not exposed to PFOA. We then looked at the gene expression of genes involved in de novo lipogenesis, or the fat creation in the liver during pregnancy. And these 3 genes had an increased expression, indicating that there was increase in the fat production in the liver, as well as the mitochondrial genes in the liver were also increased in expression in those dams or pregnant mice that received PFOA exposure, looking like the mitochondria are working extra hard to metabolize the increased fat. We also found that there were increased concentrations of triglyceride species in the liver, and some of the genes involved in processing those liver triglycerides were enriched. Furthermore, these types of fats called ceramides and sphingolipids were also increased expression in the liver of the dams that were exposed to PFOA. And this figure here suggests that the ceramides and sphingolipids are really toxic fat species in the liver that contribute to insulin resistance and the gestational diabetes phenotype in the mice that were exposed to PFOA during pregnancy. So these dams delivered, and after delivery, we assessed their glucose metabolism first at 3 months of age. And just like many humans, these pregnant mice, after they delivered, their glucose metabolism normalized immediately after birth. And then at 6 months postpartum, they did reemerge to have some abnormalities in glucose metabolism, which is somewhat analogous to women who have gestational diabetes and then normalize their glucose metabolism after delivery, but then in middle age develop type 2 diabetes. So again, the area under the curve for glucose is elevated. In addition, they have higher fasting glucose levels, higher fasting insulin levels, and then some of the mitochondrial genes are also enriched in the liver at that postpartum age. We then went ahead and wanted to see what happened to the pups of those dams. And here you can see that in both the female and male offspring, there really wasn't, at 6 months of age, the offspring didn't have increased weights or fat or lean mass. They were very similar to those that were not exposed to PFOA, but we did find that there was some fasting insulin resistance in the females. When we looked at the female, the gene expression in the liver of the female offspring, again, some of the genes related to de novo lipogenesis were enriched in the female offsprings, and this mitochondrial gene was decreased, sort of suggesting that potentially that the mitochondria were not working quite as well in the offspring, the female offspring. In summary, in this model of our gestational exposure to PFOA, the dams or the moms had a mild gestational diabetes phenotype that was characterized by fasting insulin resistance and some lipid abnormalities in the liver, and that the glucose metabolism improved after pregnancy. But by 6 months of age, or 6 months postpartum in the pregnant dams, they had glucose intolerance again with fasting insulins. And that was analogous to the type 2 diabetes in women who had gestational diabetes during pregnancy. The PFOA dose that we chose was actually a dose that was based on some of the studies that were used to develop, the animal studies used to develop the CDC health advisory for the PFOA content in drinking water that's currently listed as 70 parts per trillion. So in fact, the dose we're using is about 30% of the dose they used in those animal studies. So it's a very low dose. And then when we looked at the offspring, interestingly enough, the offspring effects were mostly seen in the females. There was a trend in increasing weight and adiposity, but it didn't reach statistical significance. We did see a fasting insulin resistance phenotype, and we did see some changes in the mRNA expressions of hepatic genes that are responsible for de novo lipogenesis and mitochondrial function. So it's more to come to think about what's happening with the offspring, but we're very interested in these effects in the female offspring. So moving on, we did a little bit more work trying to understand in a developing fetus, specifically in the liver, what could be the effects of PFOA exposure in utero. And so with this, we did a cell culture experiment using HEPA-RG cells, which are human-derived, undifferentiated liver cells, and they were treated with a dose of PFOA prior to differentiation in cell culture. And so essentially these cells were treated for 48 hours with PFOA or with vehicle, and then they were differentiated or left in the undifferentiated form. And then we looked at gene expression, and we also looked at DNA methylation. So in our RNA-seq experiments, our gene expression experiments, we looked at pathways of genes that were affected by treatment with PFOA. And in this analysis, we found that there were enrichment in pathways involved in liver hyperplasia or liver growth, or some of the liver cancer pathways, as well as fatty liver pathways, hepatitis, and fibrosis. And all of these pathways have in common fatty liver disease, which is a key component of the metabolic syndrome, including prediabetes and obesity. So this slide shows the number of genes that were affected when we were treating with PFOA and those undifferentiated and differentiated liver cells. So more genes that were affected had a decrease in expression. But one key gene that's a key gene in the development of fatty liver disease is EGR1. And here we could see that there was a significant change in expression of an EGR1, a decrease in the PFOA-exposed undifferentiated cells. We also looked at, did some genome-wide sequencing studies to look at methylation changes in the cells that were treated with PFOA. And essentially, the methylation changes could affect which cells had changes in gene expression. So what's most interesting to us on this experiment was that there were a whole lot of genes that had 100% changes in gene methylation, 100% change in methylation at specific sites, and that there were also some specific areas of the genome called differentially methylated regions where there was greater than 10% change. And we think these would be key regulatory regions for gene expression changes. When we looked at the differentially methylated regions, we wanted to see what part of the genome was affected. And similar to other studies we've done, it wasn't only the more common regulatory regions include promoters and exons of those genes, but also introns and intergenic regions, indicating that some of the former DNA regions that were thought of as junk were actually probably quite important in regulating gene expression during development. And finally, we wanted to see of those regions that are important for regulation, which are the transcription factors may have been affected. So we looked at the differentially methylated regions and tried to see what sort of transcription factors may bind there. And one of the key genes for fatty liver development is EGR1. And that's a common theme in this cell experiment that we did, was that some of the key genes that are involved in the development of fatty liver disease were affected when we did treatment with PFOA. So for this portion of our study, we found that these hepatocyte progenitor cells, when they were exposed to PFOA before differentiation, they left to gene expression changes that could be linked to fatty liver disease and specifically around EGR1. And that specifically our genome-wide DNA methylation studies did find these important regions, differentially methylated regions, that align with key transcription factor binding motifs, including EGR1. But we still need to do further studies to understand the long-standing metabolic effects of in utero PFOA exposure in the offspring. I want to end with just a few common things I often, when I present either to clinicians or sometimes talk about my work to some of our patients that are interested in these topics. It's hard sometimes to translate our findings into what people can do to help their health. And it's quite difficult. And I think that slide I showed with the map, with the updated map with the PFOA or the PFAS contamination really hits home that the exposure is ubiquitous. But in the spirit of trying to limit your personal exposure, I still think there's some evidence for the following. So trying to ditch your nonstick cookware or using lower heat. Also supporting your local regulatory agencies that are trying to limit PFAS exposure in drinking water. Many people, especially in our region, still do use private wells. So trying to work to have your well water tested so you know what your exposure is. And potentially considering installing a PFAS removing water filter. There's a number of different filters on the market, and they can definitely reduce your exposure if not eliminate it. Also consider trying to limit your takeout food and wrapper exposure. You may or may not know that some of the PFAS, some of the dental floss is actually covered in PFAS. So trying to limit your exposure that way. And then trying to consider some of your home furnishing exposures to stain-resistant carpets and furniture. Finally, if you're going to pop popcorn, maybe consider doing on a stovetop. I wanted to thank a couple people here who've done a lot of this work. Apurva Joshi is a very talented technician. She did the mouse studies, and she did wonderful work in that region, as well as some data analysis. And Stephanie Green is an assistant professor at Johns Hopkins University. And she did the HEPA RG work, trying to understand the programming for fetal exposure for fatty liver disease. I also want to thank the SEAT for funding, as well as NIHS, and then finally our Pediatric Endocrine Society Fellowship Research Award. Stevie Green was a recipient of that, and that enabled her to do the work on the HEPA-RG cells.
And it's Joanne Stanton, and she has a degree in public health from Temple University. And she's the co-founder of Bucksmont, I hope, Joanne, I'm pronouncing that correctly, the Coalition for Safer Water. And she's a community and political activist for children's environmental health issues. And she also serves on the National PFAS Contamination Coalition. She's from the area, from Warminster, Pennsylvania. And I think part of the reason that she's been really, really interested in this topic is that she grew up in Warminster, around Warminster Naval Air Base and Willow Grove Naval Air Station. So those are both national priority Superfund sites. She also co-authored a book, Behind Closed Doors, the practices harming our children's health and what we can do about it. So again, thank you, Joanne, for speaking today.
Basically, my coalition was founded to help educate area residents about exposure and health effects related to PFAS drinking water contamination. We also advocate and partner in health studies and programs for the community, and most importantly, for changes in our laws to protect the public from PFAS chemicals. Basically, I guess I was one of those rats Sarah was talking about. I grew up in Warminster, Pennsylvania, about 2 miles down the road from 2 separate military bases that both used AFFF foam, a highly toxic foam used by firefighters that contains PFAS. The Department of Defense used this PFAS foam in daily firefighting training exercises, and the foam leached into the groundwater and basically poisoned private and municipal drinking water wells, not just in my town, but at over 500 sites across the country. My PFAS story really started when I was a young mom. I was naive. I thought if food was on our store shelf that had been tested and safe, I thought if water came out of my kitchen faucet, it was safe. I had no idea just how weak our chemical laws are and how many untested and unregulated chemicals like PFAS can easily make their way into our drinking water. I unfortunately learned the hard way how important our environmental laws are in safeguarding our health, especially the health of our children. About 9 years ago, my community was devastated to discover that our drinking water had been highly contaminated with PFAS for about 50 years. For some of the highest levels of PFAS pollution ever detected in public drinking water wells. So naturally, as a mom, I started to read everything I could get my hands on about PFAS. And when I began researching the health effects, I learned that some of these chemicals can cross the placenta and affect a developing fetus. Animal studies had showed that it can cause tumors, cancer, neurodevelopmental problems, and even second-generation health effects. So the magnitude of what I was uncovering hit me like a ton of bricks. And I vividly remember the day I realized what I was reading. I fell to my knees and I started crying because my mind raced back to when my oldest son was diagnosed with a cancerous brain tumor at age 6. Back then, after my son's brain surgery, epidemiologists came into our hospital room and began pummeling my husband and I with very pointed questions. They asked, "Where do you live? Where was your early pregnancy? Have you or your husband ever worked with chemicals or pesticides?" They told us they found embryonic tissue in the center of my son's cancerous tumor. That meant it started to form during my pregnancy. There are 3 of us who grew up in Warminster on the same street, in fact, within 8 houses of each other. All of us had children of our own with brain tumors. All of our children's tumors were cancerous, and they all had embryonic tissue in the center. Doctors immediately questioned our environmental exposures, and we realized that we all drank PFAS-contaminated water throughout our entire childhood, and we drank it during our pregnancies. As a mother, you can imagine it was gut-wrenching for me to be told that my exposure might have actually caused my child's cancer. But what really sickened me was learning that the Department of Defense and the chemical manufacturers had known since the 1970s just how poisonous PFAS are. Yet they chose to be silent. They chose to watch children in surrounding communities get sick, and in some cases even die, without ever warning us. Unfortunately, the Department of Defense is one of the largest polluters in our country. And it's very ironic, I always say, that the very entity whose job it is to protect the American people ended up giving the American people and our children a lot of diseases and cancers because of the irresponsible way they handle toxic chemicals. In my town where I grew up, we have 3-year-olds diagnosed with kidney cancer who may never get a chance to do something as simple as ride a bike and put their first tooth under their pillow. There are new moms that don't feel they can safely breastfeed their babies because of the high levels of PFAS found in their breast milk. We recently had an 11-year-old girl diagnosed with ovarian cancer. Parents shouldn't have to live in a crippling fear that every lingering illness in their child will end up being a chronic disease or a cancer diagnosis that could have been prevented with more rigorous drinking water standards. My story is not unique. There are thousands of personal stories like mine across the country, stories of cancer, chronic illness, frustration, loss of income because of chronic illness, loss of home values because of the PFAS contamination, and loss of family pets through diseases and cancers as well. On a personal note, my son was actually one of the lucky ones. He survived cancer, but it didn't come without a price. As a mom, watching my bright and vibrant child slowly fade into a disabled adult has been one of the hardest things I've ever had to do. So today he's in his 30s. He still lives in a flip house. He can't drive. He probably won't marry or have children or experience many of the joys in life we have dreamed up for him. And I now realize that stronger regulations governing environmental pollutants like PFAS have actually prevented needless suffering, not just for my son, for many children in my community. Okay, I'm sorry about that. So anyway, that's my story. So now we can go to the slides, Jessica. Just to give you some, this is basically to give you some broader perspective on PFAS in our community and some of the community concerns for children's health. There we go. Okay. So we had about over 100,000 area residents in the Warminster, Horsham, and Warrington area that were exposed to some of the highest levels of PFAS, as I said, ever detected in public drinking water wells from 1970 to about 2016 when we found out. To put this in perspective, we drank it. It was mixed with dry baby formula. We cooked with it. We showered with it. We filled our pools and swam in it. And it poisoned the most sacred form of nourishment we have for our children, breast milk. For almost 50 years, these chemicals bioaccumulated in our bodies due to constant exposure. So these were our community exposure levels at the time we learned about it. Earlier this year, the EPA established an enforceable drinking water limit set at basically non-detect, under 3 parts per trillion, for 5 of the worst PFAS. ATR and EPA now feel that no exposure level is safe. And these were our drinking water levels. Worst of all, as you can see, was our military children at an on-base daycare center. Just to give you an update, obviously, since our finding out about all this, our public drinking water wells have now been remediated through granular activated carbon. They're also called GACs or RO, reverse osmosis filtration systems that have been put on our municipal wells. And they have gotten our PFAS levels down to close to non-detect. We still have over 1,000 residents with private wells potentially exposed to high levels of PFAS due to the legacy PFAS contamination with both of our military sites that's going to take decades to clean up. It's a huge problem because many private well owners have children. I mean, it's important to realize, actually also I want to say that we weren't just exposed to PFAS. We were exposed to heavy metals, TCE, PCE, hexagon chromium, dioxins, and more in our drinking water. These bases were already declared EPA Superfund sites prior to learning about PFAS. And as you know, we're just starting to learn about individual PFAS. We haven't yet started to really learn how these chemicals mix with other chemicals and what the combined and cumulative effect are on children. That's what scares me the most. We are engaged in our community with several different PFAS research projects. The one we're currently working on, my coalition, we are community liaisons for ATSDR's PFAS multi-site study. This is examining the human health effects in communities exposed to PFAS through drinking water. We are one of 7 sites across the country. Our site recruited 1,200 adults and 90-some children. We were also a pilot study for this ATSDR study. just to share with some of the investigators who might be on the call, some of our data that we collected from children that might be useful for other research includes extensive water modeling and water sampling, PFAS blood serums and urine samples and laboratory results that includes liver, thyroid, kidney functions. Also, neurobehavior assessments were done in children, as well as health histories and other surveys. Unfortunately, we have no studies that look exclusively at children's health in our community, but this is an area of great need. So, next slide. Thank you, Jessica. We can just skip over all this. We already went over all of this. Some of the health effects, we've already discussed this. Yes, that's where we want to get to. So what are some of our community concerns? Our number one community concern that you hear repeatedly, and not just in our community, but community leaders across the country with PFAS exposure are worried. Will my child's exposure result in cancer or developmental problems such as autism? We know that PFAS can cause second-generation health effects, and there seems to be a large number of residents like me who grew up in an affected area, exposed to PFAS and other endocrine disruptors our entire childhood. And we went on to have children on the autism spectrum or children with cancer. And as you know, this will not show up in local health statistics because most of the time we move away from our hometown and we start families elsewhere. So our exposure during childhood or exposure during pregnancy may have been in one zip code, and our diagnosis or a child's diagnosis occurs in another zip code. Just some little anecdotal information on our other autism, why we're concerned. At our local 10-year high school reunion, I guess it was 1995. So there was a 10-year high school reunion in 1995. The classmates got to talking and sharing and found out that there were at least 20 moms in this graduating class of 250 that had children on the autism spectrum. That year, the autism rate was 1 in 1,000. Our local high school class rate was 1 in 12 or 1 in 13 that same year. So there's a huge need for childhood exposure outcome surveys. I know that they're super expensive, but are also greatly needed to understand the health effects on children. Another concern is education for pediatricians. We are hearing, even when this first happened and it was on the news, people would go to their pediatrician's office concerned about their children, and the doctors didn't really seem to have an idea about the exposure. They didn't know what PFAS was. And now that we've had our health study in our community and we have children and families that are getting their PFAS serum results back and other lab work back, they're encouraged to take it into their pediatrician's office to discuss this. And the practitioners seem to be in the dark about the issues, the health effects and the health risks. Many of you may know about NASAM, which ATSDR had charged NASAM to come up with some physician guidelines that has proven to be hugely helpful. If you can imagine this, when this first started, we didn't even have blood tests to test for PFAS. We finally got blood testing and people were going to get their blood tests, and then there was no normal range for PFAS. There still is no normal range for PFAS. But what NASAM was able to do was they actually able to give us some type of guidelines for risks that would be associated with certain levels of PFAS found in our blood system. So this new NASAM physicians guidelines are proving very helpful to community members, but it's obvious that more education is needed, including partnering with pediatricians and advising them on ways to help us reduce everyday exposures in children. Due to lack of laws, we have many children in our community, as I mentioned earlier, that are still exposed to PFAS through private wells. Most private well owners say that they were only tested once by the military to check their exposure, and they need additional testing because there's large uncontained PFAS plumes that are still in the area. So these PFAS plumes are kind of underground, they're moving, they're shifting. There are people that have had their wells tested 4 years ago, and let's just say it was, at 70 parts per trillion. Now it's testing at 120. So it's a moving target. I also have a lot of new homeowners that are moving in. Homes are changing ownership, and there are no laws for real estate or anyone that you have to test your water for private well owners. So still is a need for education with this. And PFAS are still not part of standard water testing. So a lot of people will say, oh, I had my water tested and nothing about PFAS showed up. That's because PFAS was not one of the chemicals that were tested for. So this is specific test that you actually have to ask for. So we do know that children with private wells are at risk and need water testing. They need access to home water filtration systems. They need blood serum testing and behavioral health assessments to continue to monitor their growth, their learning, their behaviors. It's important to teach families strategies to reduce their other exposures. The new NASAM guidance calls for children with high exposure to PFAS, such as our community, to have blood serum tests and provide families with all the same things that I just described for private wells. The problem is this is all very expensive. The neurobehavioral tests for, attention deficit. These are super expensive. Blood serum testing, very expensive. It's about $150 for a home kit for PFAS to go out and just take a little finger prick and send it away. It's about $600 at a lab to have a blood test. There are many issues with getting insurance to pay for it, using proper testing codes, insurance codes. So we're currently working with Quest and LabCorp to get PFAS testing information to not only residents who are exposed, but also physicians. And we're also working with lawmakers to try to get PFAS blood tests covered by insurance. Another concern is we know that PFAS drinking water exposure in children can cause a decreased vaccine response. Moms in our area want to know if their child's vaccine is effective or not. There are parents who are actually asking their pediatricians for titer testing so they can stay vigilant if there's an outbreak of a vaccine preventable disease. Another anecdotal information is interesting. Our little coalition had a small meeting. There was 30 residents there, all with lifetime exposures. And we were discussing some of the side effects and the children's issues came up. We had 3 females. They're all working as new nurses just out of college that reported being titer tested for hepatitis due to needle sticks. And they all were found not to have any antibodies, 0, to their hepatitis vaccinations. And they had been fully vaccinated, fully updated in all their vaccines and boosted for all of their clinical rotations. So per protocol, they were all revaccinated, end of story, no more tests. They still don't know if they worked, but it begs me to ask, what is happening to our children? Are their vaccines working? Will revaccination work, or will they have the same response? And do the benefits outweigh the risks of revaccinating? So this is, I think, is an important research area. To have 3 people in a room of 30 that had this experience was frightening. Another area of concern is always breast milk. To breastfeed or not breastfeed is a loaded question for moms, especially in a community that's newly exposed. Similar to blood serum tests, you can have your breast milk tested. And we have had residents that did, and they initially reported high levels of PFAS in their breast milk. We know breast milk is best, but we question, is pump and dump a better option depending on the level of PFAS that's in our breast milk? If we had lead in our breast milk, would we still breastfeed our babies? I feel like it's not really a one-size-fits-all answer. We have one resident I know of that had over 5,000. That was the level that was found in their blood, not their breast milk. I'm not sure what their breast milk was, but there is some super, super high exposure levels in our community. And I think that when they read these one-size-fits-all, whether it's ATSDR or NASAM that comes out and says, always breastfeed is always best, I think they have a hard time with that, and they stress about breastfeeding and knowing that it might cause harm, and they also then feel guilty if they're not breastfeeding. So this is an area that definitely needs more research. So obviously there's many potential areas of concern for children exposed to PFAS, but most important is ways that you can empower yourself and you can protect your families. So next slide. Obviously, Sarah already talked about this stuff. Get your water tested if you are in an area with PFAS, either through military bases or through industry. Get a water filtration system. I just think that's the smartest thing to do. Don't even just get it. And they have these dual filtration systems that are like combo units. They're GAC and RO. They're great because each one filters out different chemicals. So, GACs only filter out these chemicals, ROs only filter out these chemicals. Some do cross over into both. but protect yourself because, you know, we're only testing for the 80 or 90 MCLs that the federal government has water standards for, and the others aren't. There's so many unregulated chemicals out there that it's just best to take precautions and protect yourself. If you do know you have high levels or exposure, get your blood test. Get your blood tested. There's blood testing. Talk to your healthcare provider. Talk to your pediatricians. There's so many preventable approaches for early detection. Just getting your children's cholesterol checked. Different panels depending on your blood tests. And lastly, politics and laws. Again, Sarah mentioned this is where change happens. We were fighting, not just me, several community leaders across the country. We've been fighting, we've been going to DC, and it's taken 10 years, and we finally got an MCL for PFAS in our drinking water, which is just a huge accomplishment. So it can be done. It's the best way to protect ourselves is through changes in laws. So I just want to say thank you all for having me here today to share my personal story and the story of our community. And thank you all for being here. Your work is desperately needed and deeply appreciated. So thank you.